Hi, everybody. I know I am in the unenviable position of being the last person between everyone and lunch, so I will be as brief as I can. I am Jackie Harris. I'm a neurologist and neurogeneticist at Kennedy Krieger. And I am a clinical translational researcher who all of my work is in what we call disorders of the epigenetic machinery, of which Nicolaitis barrester is one. So the SMARCA2 gene is an epigenetic gene. And I have only a short talk today, so I'm not going to go into what that means. Happy to talk more about it. But basically, we think that these disorders are very unique because they all pretty much affect the brain, which is what I am interested in. And you heard a lot about seizures earlier, and I'm a neurologist, so I think about seizures, but by training, I'm a neurodevelopmental neurologist, so my primary interest is in kind of higher order brain functions of cognition and behavior, and all of these disorders of the epigenetic machinery affect cognition and behavior in some way. And in addition, they are in a unique position to potentially be more treatable than other genetic conditions. And so my primary interest is in the disorders that I see to make sure that we do what we call clinical trial readiness, which is make sure that when there are therapies available, that we also have the ability to actually conduct a clinical trial. So we're going to talk a little bit about that now. So thinking about genetic causes of intellectual disability. So prior to the 1970s, there was just clinical descriptions of patients that led them to be grouped into syndromes. And these were at the time kind of thought to be untreatable. Then in the 1980s to the early 2000s came the Human Genome Project and this big boom in discovering genetic mutations that cause syndromes associated with intellectual disability. There wasn't a lot of work into the mechanisms of how do these gene functions actually cause problems in the brain. And they still were largely thought to be untreatable, but there was this big explosion of this gene causes an intellectual disability syndrome, and so does this gene, and so does this gene. Then in this century, began to be more study into mechanisms. How do these gene mutations cause intellectual disability? And people started to investigate therapeutic options. And presently, there are actual clinical trials that have begun in certain genetic intellectual disability disorders. So this is exciting. This is all forward progress. We're going to go into some of the other things that have been happening since those trials have started. So in 2011, 2012, there was this drug called arbaclofen. It showed really promising preclinical data for a disorder called Fragile X syndrome. And it entered phase 3 trials, which is kind of the final big phase before a drug can be available on market. And basically what happened is they did this trial and the trial failed. And that's fine, right? Sometimes drugs don't work, no problem, right? We wouldn't want to give anyone something that doesn't work. The problem is even the investigators of this trial will tell you that, in fact, we do not know right now today whether or not arbaclofen worked for certain things in Fragile X syndrome and whether or not that could be a treatment that could be making the lives of those patients better because they didn't design a very good trial. They didn't really think about what are we measuring, how are we measuring, what does better mean, how much better is better enough, all of these things. And so they learned a lot from that process, right? But unfortunately, what happens is if you enter a clinical trial and then you fail, by and large, that drug then disappears, right? There's no getting it back, it's gone. And so what I don't want to happen is I'm okay if drugs fail. If they don't work, I don't want to give them to patients, right? But what I don't want to happen is to go through all of this stuff, all of this money, all of this time, all of your effort, and then get to a point where we end the trial and we don't know whether it works or not, and it might be lost to patients forever. And so my goal is in making this happen. A different example is just 2 years ago in 2023, Roche suspended development of a drug for Angelman syndrome after their early phases, despite some promising results because they didn't really meet their endpoints. And again, we think that's because the endpoints were not carefully enough designed. And so all of this to say, my goal is to do research in the disorders that I study to make sure that the endpoints are always carefully designed. So at the end of a clinical trial, if it didn't work, or if it did work, that we feel good that we could say, yes, that is a true result. It did work or it didn't work. Here are the symptoms it does or doesn't work for, and here's what we can expect. So a well-designed clinical trial has to have a specific patient population, a therapy of some kind that's being tested or an intervention, and appropriate and clinically relevant outcome measures and biomarkers. So that third thing is the part that often gets left out. So how do we move towards better trials to make sure that this doesn't happen in the future? Well, we need to have a really detailed understanding of what we call the phenotype or the presentation of the disorder, right? So it's not enough to say intellectual disability, right? As you all probably know, there are lots that can mean a huge range of different things, right? That can mean someone who lives somewhat independently with supported employment, all the way to someone who is completely dependent for every single activity of daily living. And so these kind of broad symptom descriptors are not enough. And we want to hone in on the core features that make NCBRS, and different from other syndromes that cause neurodevelopmental disabilities and seizures and other things. We need to understand the mechanisms so that we can find overlap between different disorders that may have similarities, because you all know, being a rare disease, you're not the prime target for a financially motivated pharmaceutical company. But if we come and say, hey, we have these group of disorders and they have overlap and they have similarities, and here is this clinical trial we can do where it's like a basket clinical trial where we do these multiple disorders at the same time, all of a sudden that's much more attractive. We need to validate all the preclinical studies. So everything that you hear about mouse models and cell lines and things like that, those are incredibly important areas of research. We can't do things without them, but we have to make sure that the findings are translatable to human beings. We have to make sure that whatever models that we're using actually are recapitulating what we're seeing in the human beings, so we can be confident that the results are translatable. And then, of course, we need those, you hear these words again, these valid and specific biomarkers and outcome measures for clinical trials in the individual disorders. So as I was saying, these epigenetic machinery proteins, of which SMARCA2 is one, are really important for cognition and behavior, right? And so SMARCA2 is what we call a remodeler. So I have that purple arrow there. So basically what it does is it takes the chromatin and changes the shape that it's in. And changing the shape that it's in then leads to genes being more and less available for making proteins. And at the end of the day, that's what genes do. Genes make proteins, proteins carry out all the functions in the body, right? And so this is why we call it epigenetic, because it's not really the direct effect of the loss of one copy of SMARCA2 as much as it is the way that loss of one copy changes all these other genes later on. And again, as I said, this is a little beyond the scope of the talk, so I'm going to skip through this quickly. But basically what this slide is showing is that these epigenetic disorders have unique possible mechanisms of treatment that other types of disorders do not. And so then what can we do? So I talked about this deep phenotyping, or really understanding the core features of the disorder. And this leads to outcome measures. So one of these disorders of epigenetic machinery that I do a lot of work in is something called Kabuki syndrome. So like I said, it's not enough for me to just say we have intellectual disability. What does that mean? And so we did this study and showed that people with Kabuki syndrome have this really specific pattern of intellectual disability where everybody's different, everybody's along a spectrum, but okay at these things and really, really bad at these things, right? And so again, that's where we get into that clinical trial design. If we had just gone in full steam ahead before knowing this information and tested at the end of a clinical trial those things that they already do that they're okay at, guess what? We would have failed our primary endpoint because they're already doing okay there. Why are we testing these things? Let's look at the areas that they're really bad at, and let's know exactly how bad they are so we know what improvement we can expect. And then we did the same thing in a disorder called Wiedemann-Steiner syndrome, which has molecular overlap with Kabuki syndrome. And we saw a similar pattern in their phenotype. So now we have lots of things in the works where these two disorders are moving towards clinical trials together. And we're pretty confident that we could see an effect because we've done this work to look at them. So the other thing you need besides outcome measures are these things called biomarkers. So a biomarker is something that is very objective and quantifiable or measurable that could be done outside of the patient and measured accurately and reproducibly. So there are lots of different kinds of biomarkers, but basically what they do is they help a clinical trial be more successful. So for rare neurologic diseases, there's about a 12% chance of a clinical trial being successful. So when you look at everything all the way from we're developing this drug all the way to the end of phase 3, only about 12% of drugs in all rare neurologic diseases get to approval. Bummer, right? But if you have a biomarker, you increase the success probability by 20%. So you more than double the success probability. So biomarkers are incredibly important. So what kind of biomarkers might we look at for NCBRS? So one is something called a DNA methylation biomarker, which is something that we look at in the blood. And that is thinking about how that, like I said earlier, how the other genes, how SMARCA2 is turning other genes on and off. And it's very specific. And we already know that there's a specific pattern for this in NCBRS. We already know that. It's already established, published, done. The next question is, can we use that specific pattern to possibly measure change or predict who's going to do well and who's not? This is a really exciting potential biomarker. The other thing that we're doing is a specialized kind of EEG. This is very different than all the clinical EEGs your kids have had looking for seizures. This is a type of EEG we're looking at to measure learning, and we're understanding how the brain is learning. In some of our other conditions, I didn't put all this data in here because it's such a short talk, but in some of the other disorders of the epigenetic machinery, this has very, very positive effects, and we would expect that possibly NCBRS does as well. And we could see how the brain is getting better at learning as we're looking at this EEG biomarker. So, quick conclusions. Clinical trial readiness really needs to be improved for future trials in genetic neurodevelopmental disability syndromes. We need to characterize and understand the specific features of these individual disorders of the epigenetic machinery to develop biomarkers and outcome measures for clinical trials. This type of research, called clinical trial readiness research, leads to these outcomes. And some features of clinical trials, such as biomarkers, may cross over between different disorders of the epigenetic machinery, which does a lot of things. One, it allows work done in other syndromes to also work for NCBRS, right? So you don't have to reinvent the wheel every single time. And which speeds up the process a lot and cheapens the process a lot. And number two is it also makes NCBRS and other rare disorders more attractive to pharma companies and people who may have a usable drug that we could use in clinical trials. So we would like to do this with NCBRS. This is what we do. We do it in a number of disorders. We've been very successful, and we would love to have you all participate in this as well. And so we are launching a clinical trial readiness study in NCBRS at Kennedy Krieger Institute. So this study has two steps, right? The first step is, unfortunately, unlike some other genetic intellectual disability syndromes, there's really not a lot of deep phenotyping work published on NCBRS. There's some beautiful work that the foundation and everybody has done on some genotype phenotype correlations and things like that, but those are really collecting broad kind of checkbox symptoms. And again, that's not what we're looking to do. We're looking to really hone in on these brain symptoms and have a better understanding of what do those look like. And so the first thing we need to do, because before we bring you all to Baltimore to do this research study, we want to make sure that we use your time wisely, right? We want to make sure that the battery that we're testing you on is very specifically designed for NCBRS and that we're going to collect usable data towards both outcome measures and biomarkers during your trip. And so the first thing that we're doing is just collecting data that you already have, right? So what we want most of all, more than anything, is clinical neuropsychological testing reports, right? So that's what we really, really need. I recognize completely that not everybody has that, right? And so we will take what we can get. We will take developmental testing done when the kids were very young. We will take your triennial IEP evaluations for those in the United States. I just did this with a group, and I forget what it's called in the UK, but you all know what I mean. Yes, that exactly. And so those, we'll take whatever you have, but our kind of golden ticket, if you will, is if you have any clinical neuropsychological evaluations reports, that's what we need most of all to make this battery. We also need your genetic testing, right? We can't use your data without seeing your SMARCA2 variant. And then once we collect enough of those batteries, which we're hoping that today we can launch forward towards getting some of those, we will be able to launch our in-person study in Baltimore. And so what that will include is paying for your travel to come and stay with us and doing several things with your child. So we'll do the neuropsychological and behavioral testing. It'll take somewhere between 1 and 3 hours, depending on how old your child is. We'll collect blood on individuals to look at that DNA methylation biomarker. We are very good at collecting blood. We do this all the time. Kennedy Krieger Institute, where I work, is an entire institution that is dedicated toward the care of people with neurodevelopmental disabilities. So we know how to collect blood on kids who are anxious about it, fighting us, all of those things like that. So we would also be happy if you all move forward with Coriell to collect your Coriell sample at the same time as we're collecting ours. But the reason, just FYI, that we are collecting our own blood is because our goal is not just to bank your blood for future researchers who want de-identified blood. Our goal is to directly correlate what we see in that blood and the EEG that you'll do while we're there with all of that cognitive testing that we did. So we need to have the same data on the same patients because that's how we move forward towards these really good outcome measures and biomarkers. And so that is basically it. So you can reach out to us if you are interested. Our email addresses are there, and then I am here, and my coordinator, Rebecca, is right there. She's going to be the point person on this project, and we're going to be here like through your lunch break and stuff. So come talk to us. We can consent you right here, and then you can go home and collect your reports and things like that, and we'd love to receive them. And so, sorry, I went a little bit over time, but I will take any questions. Yes.
Outside the States.
Absolutely. Yeah. Yes. So it a little bit depends on how old your kiddo is. Okay. 4. So we can do it all in one day. We do a lot of, we often have very busy days on Mondays and Fridays because people come to do the whole visit on Friday and then stay for the weekend and go to lovely Baltimore or DC or whatever. But yeah, we we are flexible, but no, we do it on a working day, but it's at your schedule. We schedule it at your convenience. This is what we do all the time. And oh, also I should mention that any families who come, they can have two things. They can have a clinical visit with me. They can have a clinical visit, a clinical neuropsychological visit with our neuropsychologist who does this all the time. And you can get the full report. But if you have, insurance that won't cover it for any reason, then you will still get just a research report of the neuropsych testing for you to share with school and providers and things like that. And you can always have a no bill visit with me. It just won't be a part of your medical record, but anybody who wants an official one can have an official one. Anybody who wants one gets one if you come and participate in the research, whether or not that's covered. Same thing for international patients. I should say in answer to your question that for the in-person research part, we need, unfortunately, we are a growing team, but we are not a big enough team to have testers in multiple languages who are able to both do our research and test in multiple languages. For a variety of reasons, we don't use interpreters because it makes the data unreliable. So we do need the child who is being tested to be fluent in understanding English. I understand that some children are nonverbal and things like that, but the language that they're spoken to in must primarily be English. Yes. Hi.
So my family and I have known Dr. Harris here for a couple of years, and for the first time in our long career with our son Finn, who's 6, we found a doctor that we found that really cares and truly understands what we've all gone through as families. But so for us, this study's pretty big time. But question for you, Doc, is what is your minimum sample size to get this going?
So for the first part, I would say we need somewhere, I'm going to give a range, somewhere between 10 and 20 good reports, because the reason for the range is it depends on what age, right? We would like some younger children, but kind of the more older children and adults we get, the more kind of markers we could cross off. So somewhere between that to get it going. And then for the in-person study in Baltimore, we are targeting 25 families to come for this start. We will take more. We will take everybody, but 25 is kind of what we need to feel good about our data. Based on our sample size calculations to begin with. All right.